The target of T-cell-mediated autoimmunity is difficult to identify, owing to the nature of T-cell ligands. Although there is good evidence that T-cells are involved in many autoimmune diseases, the T-cells that cause particular diseases are hard to isolate, and their targets are difficult to identify. Also, the cells are hyporesponsive and thus difficult to assay. It is also difficult to assay the T cells for their ability to cause disease because any assay requires target cells of the same MHC genotype as the patient. This problem becomes more tractable in animal models. As many autoimmune diseases in animals are induced by immunization with self-tissue, the nature of the autoantigen can be determined by fractionating an extract of the tissue and testing the fractions for their ability to induce disease. It is also possible to clone T-cell lines that will transfer the disease from an affected animal to another animal with the same MHC genotype. This has made it possible to identify the autoantigens recognized by T-cells in many experimental autoimmune diseases. They are individual peptides that bind to specific MHC molecules. In some cases, the peptide antigen, when made immunogenic, is able to elicit disease symptoms in animals of the appropriate MHC genotype. An example of such an experimental autoimmune disease is experimental allergic encephalomyelitis, EAE, which can be induced in certain susceptible strains of mice and rats by injection of central nervous system tissue together with Freund's complete adjuvant. This disease resembles human multiple sclerosis, in which characteristic plaques of tissue injury are disseminated throughout the central nervous system. Plaques of active disease show infiltration of nervous tissue by lymphocytes, plasma cells, and macrophages, which cause destruction of the myelin sheaths that surround nerve cells, axons in the brain and spinal cord. And that's the target of T-cell mediated autoimmunity is difficult to identify owing to the nature of T-cell ligands. Have a great day.